Hello, welcome everybody. I hope you had some good sleep last night. So I'm going to welcome Jason, and Jason is going to give keynote address this morning. Title of his talk is "It's Impossible to Keep Up Career-Spanning Learning in Life Sciences." Before I invite Jason, I'm going to say a few things about him. To introduce him, I think most of us know him here. Jason Williams is Assistant Director, Inclusion and Research at Cold Spring Harbor Lab, DNA Learning Center, where he develops national biology programs. Jason leads education, outreach, and training for Cybers, and Cybers is the US National Cyber Infrastructure for Life Sciences. He has trained thousands of students, researchers, and educators in bioinformatics, data science, and molecular biology. I have actually attended one of the Jason's workshop in a long time back in 2014. So he has been doing a great job. I benefited also from that. And Jason's focus has been on developing bioinformatics in undergraduate education and career spanning for biologists. He's founder of lifescitrainer.org. It is a global effort to promote community of practice among professionals who develop short format training for life scientists. So I welcome Jason.
Great, thank you very much, Sheehan, and everybody for the introduction and the wonderful invitation to be here. It was delightful. I remember a few years back when the new award came to AgBioData to see it continue, and I could see now the vibrancy of everybody still working really hard through many challenges, and we're still here. So that's amazing. It's amazing to see it. So I'm going to, yeah, okay, we share this. I was thinking to myself, let me share the screen so we'll get that.
And
People can see the slides.
And do you want the another mic? Public one?
I've got as many mics as I know. I think everything is turned on.
Okay,
If somebody can't hear me, they'll.
give this.
Okay. I've got as many things turned on as possible. Okay.
It's really fine now.
Awesome. Okay, great. So let me try to keep us on time with just 20 minutes of this, or I should stand here. It sounded like, oh, okay, that sounded better. With this topic, it's impossible to keep up. And it's a highlight of this sort of talk I've given once or twice before. So I'm usually a couple of slides short of the full deck. And I'll try to stick to the highlights so we're on the same page here. Okay, so yes, I'm at Cold Spring Harbor Laboratory, and we all wear many hats, and I'm saying this just in case, since I'm only going to cover just a little segment of some of the things that we work on. Of course, you know, I'm part of Cybers, which we've been part of AgBioData since the beginning, and also on the board for DryApp, which is a data publication platform. We're now active in the Carbon Trees Journal for Open Source Education, so very much concerned with these areas. And I'm just putting them up in case there's a chance for us to talk later and any of those things are interesting to you. You know, like at the end of my talk, I'll have like commercials of like, hey, you're just in this one. So get your cell phones ready for the QR codes at that part. So as mentioned, I'm at the Learning Center at Cold Spring Harbor Laboratory. And so I think a number of you might have visited Cold Spring Harbor at one time or another. You may not have visited the Learning Center, which is the distinct unit within it, which works on hands-on education, molecular biology, and bioinformatics, everything from middle school and up. And we impact about 500,000 students a year in that small space. Most of them are obviously virtual or in other sites that we happen to work with. But in person, we see about 30,000 students that come to us. In fact, I took out the slide, but we have some centers across the New York area, but as far away and as large as the one that we have in Suzhou, China, or alongside the Cold Spring Harbor Courses Center there, and the newest international in Nigeria. So we have definitely invested a lot in bringing hands-on molecular biology and bioinformatics education to students at an early age. But this talk, and for this audience, I'm talking about keeping up with the future, which I think all of us are always trying to do. And we know in this audience, the future of everything we're working on is computation. That's a major thread throughout all the things that we have to deal with, the challenges that we're working with. And so I would say that my goal personally, the thing that I really work on, is how do you help researchers and educators learn how to use data and computation. And it doesn't matter what level of sophistication you're at, right? You could be a newcomer who's never coded before, or you could be an old timer and all of a sudden, some people are telling you that everything you have to write has to be in Rust now for some reason. So, you know, that's something you're always going to come across. And how do you deal with that? And really the problem, and I think the anxiety and the challenge of dealing with that comes from a changing skill set, but I would say that it's really well justified. I always use this slide as an example of the number of citations for machine learning on PubMed. And to see a hockey stick inflection in the number, that means, right, that there's new techniques that are entering our domain constantly at a rate that how can you possibly keep up with that? And so how do we think about making that scenario something that's more manageable? It's not a, you know, history is always changing, right? But it's actually pretty weird. We've seen transformative things like AlphaFold. And even with these transformative technologies, there's always that sort of hype cycle where there'll be a set of people who lay outside the domain who think the problem is solved, and the people inside the domain who are they're happy about it, but also there's still a lot that's unsolved, and also a lot of dangers, right? Because the training set that AlphaFold is trained on doesn't take into account a lot of biology that is largely unexplored, and so it's helpful, but only to an extent. And when we look at the way that we typically think about the progression of skills, I always refer back to, if you think about being at the bench, for those of you who might come, hopefully somebody from besides me, I think I see a few faces, come from a molecular lab, where in the 1980s, maybe you were doing PCRs and, you know, fragment-length polymorphisms, and then microarrays, and then CRISPR. All of those are sort of in the same wheelhouse. I've never done any work with CRISPR, but if you put me into the lab, I'm sure in a week I can have something working, right? But if you look at the computational skill set, you have people maybe like reserving time on a typewriter to type their thesis in the same room that you have people who are working to deploy something on Amazon Lightsail, because that's the way to do it. And I argue that's like a really unfair skills gap in the way that things have changed for the computational skills that you would need still to be considered, you know, I don't argue everybody needs to know everything, but clearly for a lot of the work that you want to do, you need to be able to access these technologies and what they can bring you. So that's the major challenge, right? Because educationally, the curriculum that we have at the middle school level, high school level, undergraduate level, doesn't really keep up and react to that. I guarantee you that the things that are relevant to you right now are not really what we're teaching the next generation of students. And it's like all that debt needs to get paid off in graduate school somehow, and it doesn't work. Because you're now asked to do impossible things, or you can only invest in certain people to learn certain things and not everybody to learn it. And this kind of leads to sort of a scientific digital divide. I don't want to misappropriate the term, but the term originally is referring to groups of people who don't have access to the internet or don't have access to things. But I think as professional scientists, you actually could be in a divide where there are groups of scientists who know how to use the thing and groups of scientists who don't know how to use the thing. And it's very hard to figure out how to get from one place to another. Some people might be positioned at a research institute like Holsworth Harbor or a large enough university, or at least amongst your colleagues, you have those people, or you have the resources to hire graduate students and postdocs who may bring those skills into your lab. But you could just be a plant scientist sitting somewhere, or you could just be somebody that's working on animal genotyping somewhere with a really interesting scientific question, but it's just you who's going to be the person who's responsible for moving that research question to the next level. And it's a question of, do you have that? There's a comment.
Oh, just somebody online said the sound stopped.
Okay, I'm still broadcasting, I'm still speaking. It says still talking, Sunita. Maybe they'll realize. Okay, yeah, they just muted themselves or something. We don't want them to be divided out. We want them to hear something. I'm almost done. And interestingly, you always hear this problem on a social level, and I found this interesting myself a couple of years ago when I started the project I'm about to tell you about, which is, yes, it's all really similar. This was a paper, everybody's heard that we don't have enough STEM skills in the country, there's not enough STEM workers. But this is an analysis, National Bureau of Economic Research, where they basically said there's no shortage of STEM workers, it's only a shortage of STEM skills. And what happens is that people follow a career trajectory, and then they reach a point at which it's not possible for them to get new skills because they've got to do the work they have. Nobody usually pays you to go off and learn more things, even though we have these ideas of like, oh, take a sabbatical, but that's a limited use. And this idea of in STEM, the skill set changes so rapidly, it's a real big thing. We had done a survey years back when we were talking about NSF funded investigators and asking what were their top needs for dealing with big data biology. This is published in PLOS Computational Biology. Sorry, this reference is small, but all of the top needs were in training and not necessarily on bigger hardware. So that was there. Another study we did looking at the undergraduate curriculum showed that most faculty think bioinformatics should be a part of what they're teaching, but very few do it. We're working on manuscript. We redid the study and went to other groups and started finding the same things. In fact, still holds true from unpublished data, but from what we published before, that the newer faculty members who are trained are the least likely to bring what they train into the classroom, right? It's actually hard to change a curriculum. You're not rewarded for bringing in new hard things that students give bad evaluations on because it's hard. And as you're maybe tenure, you're well supported, but at the same time, also, we don't know exactly, but as the techniques become harder to train on because it's so complex, how much of that can you really bring to your students? And this is it becomes an equity issue because again, it'll matter a lot. Where did you end up with? Are you starting off at a point at which you have a lot of resources and you can make up some of those differences, or are you starting from a point at which the rich get richer and those who are poorer are not going to get ahead? So how do you solve these problems? Well, from my perspective, or the tool that I have, or the thing that I'm responsible for, it's how to improve the quality and the application of professional development. Because we come to even a conference like this, although it's a lot of dissemination about what's the current work, what are the current challenges, we're also here to learn from each other. And sometimes we explicitly come together to learn, to take a course or look at the material, follow a tutorial, try to get something. I can't tell you how many machine learning tutorials I've emailed to myself, like, I should take this. And very few of them end up even getting past the logging onto it. And a couple of years back, there was this paper by Feldman et al. PNAS on the null effects of boot camps and short format training. And they did this longitudinal study of several hundred graduate students, following them for years across many different institutions, and finding essentially that they couldn't tell the difference between graduate students who've taken the interventions like a coding camp, a boot camp, a workshop. And so that's disturbing because if we spend time doing that, I spent a lot of time doing that. And so why is that? It doesn't mean that all training isn't effective, but it means that oftentimes when we come together to learn about a topic, you have a number of problems that could arise. One, it could be that the person that's teaching the topic is an expert in that topic and has no idea how to teach. And so you are sitting there desperately knowing that this person is really, really good at what they're talking about, and none of it is coming across. And so you're hoping by diffusion that somehow it makes it across. But it's a big challenge. That's one of many problems that can happen. It's a big challenge because we spend a lot of money on training, and we write grants all the time saying that, oh, after this is done, I'll put a tutorial up about it, or we'll make this accessible. And of course, for many of the people in this room, many of you are resource producers who hope that users will come to the resource that you produce and be able to effectively do better science, and then cite you because they use your resource. So what we've been working on for the last 2 years is a minimal set of principles, and also I'll talk about recommendations for effective, inclusive, and career-spanning learning. So we were funded by NSF, as I'll mention there, to gather together groups of large training programs across the world that do training. A lot of it was in bioinformatics, it's quite relevant to life sciences, and asking the question, What do we know from education sciences about how training could be done effectively, even in a shorter amount of time? And what do the most successful programs do? What did they do right? So that we could try to take that knowledge and codify it into some principles, into some recommendations, because there's really no journal for this, and there's not really a professional society, so to speak. So a lot of these ideas sit in the place where they are and don't move further out. So you probably know courses that you've taken that you would send your students to, or you'd recommend to your colleagues because they were super effective. And there's some kind of magic. It's not magic. They've been doing it a long time, usually. But then you've also taken workshops, so you're like, oh my gosh, well, the coffee was okay, and I saw my friends go like, but that was just about it. And so putting it into a couple of principles that's worked before, if you write 10 things on a tablet or put them up, people can digest about 10 or so, although we did 14. So my slide is a little stretched out, but when we came up with this construct, you've heard of hair principles. We have the bicycle principles. Let me do that away. And the bicycle principles, despite the fact that they all end in E, I'm noticing, there are two sets of principles, which I'll talk about, and you'll see why the bicycle metaphor was useful. So the first set are four things, four core principles that any short format training could follow. The first one is start where we want, but it's a cycle. This idea of best evidence, that when you're actually designing a training course, you really need to ground it in what we know from learning sciences. And there are actually people who study this for a living. They're never listened to, but they exist, and they have recommendations and principles so that when you start designing a course, you're not designing it saying, oh, I think they should learn about X and Y and Z, and not really actually saying, well, how do people learn about X, Y, and Z? You can't learn about Z until you learn about X first. So thinking about that. How do you make sure that your training is effective? We often don't assess our training on a short format scale because who wants to take a quiz? But actually, we don't usually go into really what's effective assessment to demonstrate to the learner that what they have taken actually has moved the needle for them. We also think about what we call catalytic learning, which is really admitting at the beginning of most short format training that I'm not going to be able to get you exactly where you want to go, but I can prepare you for the self-learning that you need to do once you leave this half day or this full day that can set you up to be more successful learner on your own. And then inclusivity. How many of you have put together your training and thought about, is this accessible to people with different abilities than my own? Are there certain groups of people that I'm excluding because I'm only holding my workshop and training at hours of the day or days of the week when caregivers probably won't be able to make it? There's another set of principles called the community principles, which are much shorter words, so they don't stretch on my screen here. Reach, scale, and sustain. And those principles only apply if your training is meant to scale out. If you're not doing one-off thing that's only meant to be done once or twice, but you actually want to do reusable training, there's a set of community principles. So all of this, you can go to bicycleprinciples.org, and you will find there, as my microphone is moving, so I'm never knowing, but I guess you still hear me. At bikeprinciples.org, it's a set and an explanation of the principles. There's a video, and along with those principles, I think if I hold my shoulder the right way, it kind of picks up, or at least I get the echo. There is a set, there it goes, of an explanation of all this and all the people that did the work. And there's also a preprint that just went up a couple weeks back on bioRxiv, optimizing short format training, international consensus on effective, inclusive, and career-spanning professional development in life sciences and beyond. And again, what it's an attempt to do is start to lay the groundwork for groups of people to come around the challenges, because essentially short format training is just one of those tools that it's always going to be around. It is convenient. It is, you know, for me, I will not show up to the 10-week-long Coursera sometimes. But if I can make room on my schedule for 3 days to learn a new topic, I want to make the most out of it, and it can be an effective tool. There are a whole bunch of recommendations, which I'm going to wrap up now, but just to give an example, the recommendations are abstract. And so to keep people, usually if I go through all 14, which I'm definitely not going to do here, I always I've made use instead of surfing the internet or fishing for random photos that convey a concept, is just use generative AI to produce random images that kind of give me an idea. The first of the 4 recommendations, which quickly mentioned, is that we think that the community as a whole should work on providing professionalization for people who do short format training, so that if you do intend to do workshops on the regular, there should be tools and approaches to help you become more effective at that. The prompt is always underneath the picture, so you can tell if it did a good job or not. The second one is the idea of making inclusive practice and short format training ethical responsibility, right? So it should be ethically required that you make your materials accessible. We don't want to forget that, so we have to make it important. The next one is about using training to counter inequity. So you have to judge there, justice, something like that. It's not copyrighted by Marvel. But that's the idea that when we think about how we direct resources in a university, are we directing them towards the R1 or in this crowd where we're starting, let's say, the ag schools or the HBCUs, right? How do we direct the training to the people who could most benefit from it? And the other one is this idea of badging. When we do produce training that's of quality, how can we announce that to the public, and how can we ensure that the quality of the training that we produce meets a certain level? Could you say that, hey, this workshop is badged for accessibility, it's badged because it has learning objectives, and et cetera, et cetera. So other quick things I'll, like, again, wrapping myself up here so I don't go over. We also have to build other infrastructures that help support this type of research and education. At Cyberse, we've been doing this for a while now with our foundational open science skills courses. And I think Cyberse is not alone. I think every single one of our infrastructures, for those of us who can produce, realize that it's not just a training on how to push button A, B, or C, but it's a set of tools and approaches to working with data. I know there's a lot of talk about FAIR, right, at this conference. Those are all things that you need to get into the mindset of your users so that as they're engaging with what you have to offer, it's not just about a simple mechanical thing that they need to do in order to produce a result, but really thinking about how do they become responsible scientists working with data. So you have to build not just the sharing of your ideas, but actually building communities around them. One of those that was mentioned is a group that I've been involved with starting called Lifesite Trainers, realizing that there was no community globally for people who do training. So it's an online space, it's a Slack. We usually have calls every month on topics that are relevant. One of the last ones, although my video doesn't load, was a discussion that we had on ChatGPT. How will that affect training? I was blown away. I was setting up a Jupyter Notebook for some students, and I said, oh gosh, I have to set up a blast database. I haven't done this in like 10 years, and I want to read I don't know if anyone from NCBI is here, but you know what their manuals are like. So I was like, I don't want to read the manual. And I asked it how to set up a blast database, and it just said what I needed to do. This is going to be really, really interesting. So there's a blog post coming soon summarizing that discussion, because I don't know what training looks like in 5 years. Do we actually have to teach the AI to be a better tutor? Can it assist us in certain ways? Will it be the way to do documentation in the future? I think it might be. It's very interesting. So this is a photograph of a lot of the people that were involved in that working group to assemble the paper recommendations, and you can find all their names on the list. And at my closing slide, or my commercial slides, are just a quote by Alan Toffler. Alan Toffler said that, that's what it's going to mean to be literate in the 21st century, because we're going to have this constant cycle of unlearning things and relearning things. That's going to be so much more important than just what we think about the basics of reading and writing. And for the things that you missed that we didn't have time for anybody that's interested, come join our Nanopore network. We're putting together a whole bunch of materials and resources for people who are interested in doing nanopore education. And this is collaboration with CUNY and Spelman and the University of Puerto Rico. You can find more about that on CUBE, since we're talking about education today. Go visit the DNALearningCenter.org website and see the barcoding programs. These are all really great introduction for students. When you have students publishing, records on GenBank, that's great. Now maybe they're going to come to AgBioDatabases next. These are folks thinking about that. I think I have a commercial here for a biotechnology summit that anybody's interested in. You can see me, or you can find the slides. I guess you have them on the shared drive if people don't have access to. I think that's my last commercial. So I'll just back up to the thank you slide and take any questions we have time for. Thank you. I'll repeat any questions so that people online can have it, or maybe they'll hear the
Exactly.
Okay,
You know me, I talk about
okay, never do.
it. So do we, with the principles of what you have for bicycle principles, trying to make everything really inclusive. The majority of us try to make either both a text format for our tutorials and then a video format for it on YouTube. Are we thinking, picking your brain?
Sure,
What can I do something else? Because like, I know, like, there are people who don't have good internet,
Sure.
should I do a podcast?
sure.
But
Sure, so I don't have one answer for that. And in fact, what our next step would be doing things is sort of like putting together toolkits. But I have two things I'll say. Number one is I guess as the name implies, this idea of universal design, which I think some people are familiar with, certainly in education, which is even when you're building your slides or you're putting in your videos, your choice of fonts, your choice of contrasts and colors, all of those different things are really, really basic principles that when I'm just designing a course and I'm just trying to put things on the slide, I might not have those in mind, but there are a lot of universal design features and the whole literature in the education space on how from the beginning do you design something that could work for everybody. But then the second more important thing is, how do you know your audience of learners? In some case, you may know who your audience of learners are. Like if your learners are other bioinformaticians, they're probably going to be comfortable in the space of working with, command line stuff and reading text and things like that. But providing avenues for people who choose to identify that they need in a combination or not, but ideally you work in a way that people can have that choice for themselves or not to reveal something or to share something, because actually it shouldn't just be about why didn't they ask me or why didn't they identify themselves as needing something. So it's working with people as you go along, or as communities get more sophisticated. You start to learn from your peers and from people who are willing to share their perspective or their thing. What can you do better? And making sure that those insights are shared, right? It could be people who are neurodiverse and they don't want the videos longer than a certain length, or they want them labeled in a certain way that makes it easier for them to navigate. So it's accumulating those things and being mindful. We'll never be perfect. We'll never get everything right. But as long as we signal to people that the door is open and we're interested in that, that may be the most important thing, to signal interest that yes, we want these materials to be accessible. Here are the steps we have taken to attempt to do that. If that does not suit you, here are the ways that we potentially could make it better, or that if you'd like to give us feedback. So signaling that you care about that might be the most important thing, even if you get things wrong.
Okay, thanks.
There's another question.
Actually, I have two, but
Okay.
I'll start with the original one I was thinking about, which is, I assume like going into a classroom, like, nobody comes in as a blank slate. So I'm curious about your thoughts on like, how do you go about sort of assessing the knowledge in the room, and how your teaching sort of evolves to respond to that, like, oh, this is what you think there is. So let me go back and like rethink, for example.
Excellent question. Yeah, excellent. Because as we get, when you do have kids, at a certain point, they are blank slates, and they remain that way for a surprisingly long time. But as you have adults, right, everybody's coming with some prior knowledge. And so the strategy that I would say people like to think about is sort of, in the technical terms, metacognition, getting people to be aware of where they're at and how they might assemble that knowledge, but prompting them to say, hey, if you agree with this statement, or not necessarily, doesn't always have to be like a quiz, but you prompt them with like, hey, if you're able to do this, or if you've read this paper, or blah, blah, blah. So providing cues to people of, are they going to come in with the same page, or there's some basic things that they might want to get before they take your training to try to give people. But ultimately, in the training, it comes down to equipping people with the ability to ask the question, do I understand this? And if I don't understand it, where am I going wrong in my understanding? Like helping people to have their own tools to self-diagnose what's wrong. So if somebody's playing an instrument for as a professional, if you've played it for a long time, you do get to the point at which you don't necessarily need your teacher there all the time, because you realize, okay, I'm a little bit sharp here, or I'm a little bit flat, or I keep grouping this rhythm wrong, or something like that. So what you want to do is think about your training activities not as just blasting facts at people, but sort of saying, okay, well, I say that this needs to be findable. But you prompt them with questions that guide them to ask, like, what does it mean to be findable for a data set that's 2 terabytes? Okay, well, now in this case, here are the sets of things you need to think about. So it's more about just getting people in the minds, rather than telling people fact 1, fact 2, trying to tease out in your questions and assessments, things that prompt them to actually think and reflect on what they're doing. And if you get people in the habit of doing that, you also preparing them for when they go to new knowledge. It's a long discussion, longer than we have time for, but it's more, it doesn't mean radically changing your teaching, but it means really engaging with the questions that you ask in order to elicit them to self-diagnose where they might be with everybody else, or where they might need to be able to acquire a new skill or piece of knowledge they're missing. And if you get people thinking about their own thought, then that's metacognition, and that's the tool that people need in order to be able to grow with that knowledge that you're trying to impart.
Great, one comment, Caroline.
Yeah. There is one comment from one person online. In my experience, intensive training is great for informing people about the availability of tools, but only repeated and relevant use of those tools results in truly trained individuals.
Yes, I agree with that. Absolutely. I think the thing one of these ideas that's elaborated on the website, and I don't have time for this idea of catalytic learning, is people do have the misimpression, and even not just the learner, but the educator, that coming to this course is all you're going to need. And that's rarely the case. It's more about telling people, here's what I can give you. Like, usually all I do have time is to tell you, here's something that might be good here. Here's a new something else that you haven't heard of yet. And here's why I like it. But at least setting people up to realize that you're going to need a whole lot more than what I give you. Because what happens? You do 2 days of RNA-seq, and then people think that, oh, well, now I took that little data set that you gave me. Now when I go home and press assemble, first thing, it doesn't load. Or the assumptions are completely wrong for the type of situation that they have. And if you've taught them to imitate you, then of course they're going to fail. In fact, some people will now stop trying, although usually you're so desperate, you have no choice but to find your way through, versus saying, here's the reason why I made this choice in this tool set. Here are my assumptions behind it. Here's where your assumptions are likely need to require evaluation. So being more intentional about the fact up front that I'm not going to be able to help you as much as you might think, but here are the places here's what I can get for you. Here's what you're going to need to do on your own. You're going to need to do this on your own. I don't, even though I'm being very blunt, I don't think you hear a lot of, this is for you to do by yourself. You got to do this on your own, and just have a realistic expectation. But here are the tools that will help you once you get there. So that's probably all we have time for.
Yeah.
Thank you.
Thank you so much.
Yeah, and now we have some presentations. So the first presentation is from the Data Federation Training Group, and Peter Selby from Cornell University is going to talk about Data Federation Training Group, updates and vision for the future.
All right. Hello, everyone. So I'm the chair of the Data Federation Training Group, and I'm gonna talk to you about our working group. It's new this year, so we're sort of just getting started. There we go. Yeah, so here are our members so far, 13 of us. I think, same with everyone else who's reported so far, we're having some issues with consistent attendance, but I think everyone on the list has attended at least once or twice, so far so good. Yeah, if you hear the rest of this talk and you're interested in contributing to this working group, reach out to me, reach out to Monica, and we can get you set up. Okay, so the objectives of this working group. So I think Monica reported yesterday on the original Data Federation Working Group that happened last year. One of the big things that came out of that was the survey we did for the community, trying to figure out what people's needs were. And so then the proposal for this working group sort of was built out of that. And yeah, we can see roughly 1/3 of the Data Federation respondents would benefit from some kind of training on a lot of these technologies for data exchange and data linking, data federation type technologies. So that's what prompted this group to start. And so a lot of us from that old Data Federation group just rolled over into this new group. And so, yeah, that's our objectives, to try and put together this repository of training material to sort of catch people up to speed on what is this Data Federation, data sharing stuff all about. Yeah, so I included this slide that hopefully you saw yesterday, just as a repeat of the results from that survey. We had a number of technologies we identified from the survey that people were most interested in. For our group, we actually widened the scope just a little bit, just to have sort of the full range of technologies that we're looking at. But yeah, this working group is driven directly from the data we collected last year. Okay, yeah, updates. So far this year, first of all, we found a meeting schedule. That was no small task. And we do meet every other Friday at 12 Eastern, a successful bullet point number 1. And yeah, we developed a short-term strategy going forward for the next few months. And yeah, we created that list of technologies that I talked about earlier, that we really want to focus on. And then we were sort of discussing as a group and saying, okay, well, yeah, we'll start putting together training material about this. And we looked at each other and said, wait a minute, we don't really know everything we're talking about. So our first step was actually to try and get some experts in some of these technologies that we're interested in, get them to join our meeting, talk to our group, and sort of do a little bit of teaching the teachers type work. And on top of that, it means we can also record those sessions. And now we have recorded introductions to each one of these topics that can get rolled right into the eventual repository of training material. So yeah, so far we have had 2 presentations. I gave a presentation on BREPI and the BREPI project. Mark Wilkinson gave a project on RDF and some of the work around that his group is working on. Coming up, we have a presentation from Neuralp Merchant on the iRods technology. Sometime in June, still figuring out the schedule. Cyril Palmier is going to come and talk to us about his work with the BidAir project, which is a Elasticsearch index-driven search tool. So we'll be able to learn some of that. And I think we've got people in mind for the rest of these. We're still figuring out the schedule and trying to line up speakers. But yeah, so the idea is hopefully we'll get through this list of technologies, have a solid introductory recording for each one of these, and then we can start building from there. Yeah, so sort of our future vision as a group, looking forward to what we're trying to build, is this repository of training material. This is a perfect segue from Jason's talk in terms of having a number of different types of material, and that'll actually go into the questions we have later for the working group discussions. So it worked out perfectly. Yeah, so we're planning on having these recordings as well as sort of extracting some of the written material that we got just to, sort of introduce some of these topics. And then a lot of them have their own training material that we're just going to link to and say, here's an introduction to, where, some of these technologies that you asked about, and here's their group to go train with them more if that seems interesting. We've also talked about putting together a technical demo of at least one or two, depending on time allowed, of some of these technologies that we're exploring, and try to really illustrate some of the data sharing and data federation use cases we can come up with as a community, and try to really explore those a little bit. And then really the other key thing we want to do here, and it's something we ask each one of the presenters that we're bringing on, is trying to do an assessment of the different technologies we're exploring and the use cases that they're effective in. And so we want to try and document some of the example use cases that fit with each technology, so we can sort of advise the community and say, if you're coming to us with this type of data sharing use case, probably use this technology. If you're this other use case, use this other technology. And on top of that, some of the discussions yesterday were around communicating between working groups. So we definitely want to touch base with the education working group at some point. We've got a full schedule of presentations coming up, so we'll find a good meeting time somewhere in the middle, but yeah, we'll definitely want to touch base there. Yeah, and then our discussion questions for the breakout rooms later. Talking about data types. So as a database maintainer, we're really interested in a couple of things, was sort of what data and data types your database holds that you feel is common between many of the other systems within the AgBioData community. Something that we could develop a standard on, or use an existing standard on, as well as some data or data type that is unique to your system, but others would find useful. So that's another instance of data sharing where you've got some very unique thing that other people want to use. As a scientific user, looking at sort of the same question from the other end, what are the types of data that you're having trouble accessing that would need some technology intervention to assist with that? And then, yeah, we're asking what are the best mediums for this group or for this community? What would be the best way to present this information? We've got a few ideas, but we'd like to hear more about that. Yeah, and then along those same lines, who is the audience for this training? For those who answered the survey, who in your group, what's their role that would be most interested in taking this training? So I think that's all for me.
We have time for one question.
Okay.
I think we will think about questions in the coffee break. Okay, so we have the second presentation from Meg. And it is about the education working group, learning how to be fair, open source education tools for AgBioData.
Did you do the questions for our succession?
Yeah, okay.
This was fine. Good. Okay, thank you. Okay, so this was one of the main aims of our grant. We really wanted to create some educational material
And we really wanted it to be able to be incorporated in various types of educational settings. And so our task was really to think about the purpose, the structure, and the importance of GGB databases, GGB being genetics, genomics, and breeding. You haven't already heard that acronym 50 times past 2 days. But also really trying to help people figure out why these matter and how they benefit the entire scientific community with public data deposition and integration. They benefit both the people depositing the data and the people using the data, which is sometimes the same person. And how those databases are really essential to the fair data ecosystem, which is sometimes overlooked. So we started out as a committee to define our stakeholders. Who do we think really cares about this educational curriculum? Who will utilize it? Who will advertise it? Lots of educators at the university level, I think, are very interested in incorporating this into their coursework, talking to their graduate students about it, even research PIs at the university level. Databases themselves have a vested interest in making sure that people know about them and why they're important. And funding agencies, they want people to know where to put their data, how to make it fair, how to do the right thing. So those are all of our stakeholders. Also, there's a lot of people who benefit from the knowledge. Researchers from high school to undergraduate, all the way up through postdoc and even PIs. Educators who want to convey this material, and students. So that was sort of our very large audience. This grant landed in the middle of a pandemic. So we thought, hey, we're going to need multiple modalities. We do feel that there's a lot of benefit to that in-person interactive experience. So we want our materials to be able to be used for that. Slides, activity descriptions, discussion prompts, all of those are being incorporated into our modules. But we also want to have this kind of self-directed asynchronous modality where you could watch a video on YouTube, go through some activity descriptions and kind of do it yourself without somebody standing in front of you telling you what to do, and do some reflection exercises. So we really needed to focus our scope. We're a relatively small committee. We all have many time obligations. But one of the things we immediately noticed is there is a ton of fair data curriculum already out there. Every conference you go to, people are talking about FAIR. There's tons of modules, places to go learn about FAIR. But what we really thought the gap that we could fill is thinking about the database aspect. It's really less well covered. It's not discussed. It's like, oh yes, you should put your data in a database, but it's not really like, what are the different types of databases? Are some databases better than other? How do you pick a database? So we really thought that's where we could fill in some gaps. And this is particularly thin for agricultural resources. A lot of the FAIR data is very broad. There's educational materials, but we wanted to build something that really speaks specifically to biology and even in a more narrow scope agriculture. If you are looking for a FAIR data curriculum, FAIR has a really lovely white paper out with lots of curriculum in it that we thought was great. No need to reinvent the wheel there. So we have 7 modules in our current curriculum. We've gone through a lot of revisions and things moving around in order and getting merged, but we're pretty happy with where we've landed. We start with this really big overview. What is a biological data repository? And thinking about, and even just defining the language of data portal, database, website, data repository, data lake. Like, what are all these things? And those boundaries are very blurry. So we kind of go through some of that. And then we talk about how databases support FAIR, why they're important to FAIR. We dig in a little bit more specifically on biological databases, types of data, how to find and obtain data. There we move into data management plans. Lots of researchers are interested in how to write really good data management plans. We also want you to follow up on your data management plans and do what you promised you would do. And the trust principles, which, if you don't know what trust is, transparency, responsibility, user focus, sustainability, and technology. So this is really a framework for how do you pick a database that you trust, that'll be around for a long time, that's doing the right thing. Submitting data, how to submit your data, where to submit your data, how to use your library resources. We have really benefited from some really spectacular librarians in this group who really helped us to think about how libraries play an important role in databasing, in FAIR data. And finally, thinking about databases for agriculture, our group of AgBioData databases, what do they offer? What can they be used for? Do they have their own tutorials or training so that you can move seamlessly from this kind of generic curriculum into something specific to your crop or animal or microbial system of interest. We have slides drafted for 7 of the 7 lessons, videos completed. This might actually be 3 of the 7 lessons by now. So I think I forgot to update the slide. So we're getting there. We have material. Future additions, we're hoping that during the working group today, if anybody wants to contribute, there's a ton of great working groups here that you probably all could produce a module about what you're doing. It would be really interesting. So we would love for you to think about doing that. I think most of our modules, our videos are coming out in about 30 minutes. So it's not, we're not shooting for some really huge, overwhelming thing. It might be something that you find reasonable to put together. We thought it would be really interesting to have a little bit more background on technology of databases. What is SQL? What is a NoSQL database? How does the web really work? Because a lot of people don't know that, and it's kind of interesting. Helps you think a little bit more about how you interact with databases. Ontologies is one that we have not tackled. We have a few slides on it. I think it could be its own module. And any other community recommendations for what you guys would like to see. In addition to this, we're putting together a white paper. It has a somewhat long and unwieldy name. We probably need to work on this a little bit. I'm not great at naming things, so anybody who has clever naming abilities, please, please help. But basically, we want to do the who, what, why of agricultural databases, talk about them being essential, and then present this educational set of modules as a framework, and have real competence profiles for high school, bachelor, master, and doctoral level learners, and have official learning outcomes so that as we continue to maintain and build these lessons, we know what we're aiming for. So good pedagogy. During the working group, we would love to know if you'll use this, how you'll use it, how can we help you use this, what formats are easiest to use, video lectures, lesson plans, slides, are we on the right track? We think we are, but if you have other ideas, we'd love to hear them. Places we can get the word out and what comes next as far as sustainability, maintainability. These are living educational modules that can't be static. We don't want them to look exactly the same in 5 years. So starting to think about that sustainability aspect is really important. And of course, if you want to contribute, we would love for you to. Coming up, starting toward the end of year 2, moving into year 3, another one of our grant goals is to generate a more practical guide for fair data management available to all agricultural scientists. And we're kind of thinking about this yesterday. It might be something where you take an intersection of your species of interest and your data type, and it tells you all the places that data could go, something in the fair sharing, RE3 kind of realm, but maybe more tailored for agriculture. Because I think a lot of us from databases hear a lot that people just don't know where their data should go. So trying to create that guide that really helps them figure that out. Yeah, so we'll be looking for people who want to join that working group. So get ready for that ask and consider it maybe something that would be fun to work on. And I want to mention that we do have great representation from all over the US. We currently don't have anyone internationally. We would love some international participants. And we have a really great representation of our working group in person here. Everybody highlighted in yellow is here, so you can come talk to all of us. Very, again, wonderful group, diverse group, all the way from graduate student, librarian, professors, database people. So it's been really great. Yes. New members welcome, students welcome. And we also don't have a ton of representation from like the livestock community, so we love that. We will be having a short session at the plant biology meeting this summer. If anyone is there, please come see us. And with that, I will take any questions. I'm both the question runner around or center, so I'll try to do both.
So I would use the parent and database module in my class at UC Berkeley, and they were great. The slides were great. So thank you for doing this.
Yeah, I'm glad you use it. If you have feedback or if you have feedback, we would love to hear it. Anybody else with questions?
I'm curious, since we've seen several aspects where people are having, training in federation and some of the things that the genetic variation working group was doing, come with some, training on how to use these templates. Have you seen those training activities dovetailing with the education portion, or do you see it as a totally separate aspect of it? You ever?
Yeah. Good question. I think we're really gearing our material toward students who will be depositing data, who may not be ready for, like, let's build a API. But I think there's this great overlap. Please write an introductory module so that then when they come across an API, they know what it is and how to use it. But then maybe there's the advanced mode, which is I'm a database developer and I need to pick a data federation method. So I think that could be like intro and then advanced.
I'm a member of the working group and I can say that like a lot of the documentation is like how to use our thing, not what is our thing and why you should use it. And so this is kind of filling in some of those gaps is what I would say.
Okay. I'll head over here. I think there's some questions.
Well, just a comment. So I'm going to ask a question and part of it is also came in the June. And the idea is that as a training and questions about the fear is that fear is right now a choice and we encourage people and it is not a law, so some people think that should everybody comply and we wish so. So what do you say about that?
So fair is not really a choice if you have federal funding, right? Like you had to write a data management plan, presuming it was reviewed. You're supposed to be complying with it.
How about the patents and other type of curricula? If people want to file patents and.
Right, IP is something that you would have had to address in your data management plan.
Fair is not open. People confuse these things.
Yes. Yeah, and the FAIR framework can embrace intellectual property and privacy as well. Which is something that we should probably explore. We're definitely coming at it from a very open science perspective in our current educational models, which may be something we need to think about. But yeah, I actually am really excited that a lot of the graduate students that I have really actually want to do the right thing and are buying into the global scientific knowledge base and how we can all help each other. So I'm really excited that, at least for them, I think they're really buying into this. It's not something that they're being forced to do, but something they actually want to do. And if you give them the reasons why, they get really excited about it. So I think somebody just has to sit down and tell them, yes, open data helps everybody to move faster and helps data. Data is expensive. Let's all use it and get the maximum amount out of it and help everyone around the world. And I think that resonates. So maybe we're going carrot more than stick right now.
Have you thought of like organizing like existing educational material just for agriculture? Like yesterday I was like showing, these are like two different links where you can learn about the elixir handbook.
Yeah.
Like existing material, just organized by topic, what you need to do, this kind of thing, or like the VCA validator, there were two tools, like GitHub repository, like just collecting that all for archiving.
Yeah, actually, Anna-Rita just created a Google sheet that has anyone can add links. So we want to capture as much of that as we can to get people steered down the right path for further learning. So I need to find a way to share that with this whole group. Okay, so Anna-Rita's gonna share it on the Slack channel. I went through it. I've only added like 2 of the TARE resources right now, but we have a whole, I have like 87 tabs open on my browser and about half of them are things I want to add really soon. So it's coming. All right, any other?
I have just one comment, so just one comment, according to the breakout room that we had yesterday for the diversity working group, that I think we should also consider like providing in-person education workshop to reach out to specific and represented community and to build this relationship with these communities and something that we should consider for future plans.
I think that's a great idea. All right.
Okay. Thank you very much. Thank you.
Yeah, online, okay.
So next we have Kapeel Chougule from Cold Spring Harbor Lab. And he will be talking about gene and genome nomenclature group, current status, challenges, and future direction.
Oh, hi, everyone. Can you see my screen and hear me?
Yeah.
All right. Thank you.
You may wanna do a slideshow. Yes.
All right. So I'll be representing the gene and genome nomenclature working group. So I think we have been working on this working group since past 1 year now. And I think we had made some progress, and I'll be updating you on the current status and where we are going forward with this working group. So initially our motivation was how we identify or the importance of having accurate and persistent identifiers for genome assemblies and the gene models that are out in the public domain. As you can see, we have a lot more genomes coming, and the annotations that are associated with those genomes, but there is a lot of inconsistency of how the identifiers are being provided to those. So our motivation was, how do we provide very accurate and persistent identifiers for those genome assemblies and the gene models. So once we have that kind of working, that would help the users identify or understand multiple assemblies that are there and the annotations that are coming out from them. And it will help them also replicate results and also compare gene models across the different assemblies. And so that was our motivation, and we wanted to have something that we can work with the community and also provide as a framework or a guideline kind of a thing that we address this challenge. So initially, we kind of looked what's out there. That is, what are the existing genome assembly naming conventions. So as you can see, there are different components for a genome assembly naming. So you have the species identifier, the assembly, the cultivar, and you can have the sequencing consortium name also identified within the nomenclature. Here's an example of the Darwin tree of life, where you see you have the assembly version in orange, and then you have the individual, and then you have the the tree of life identifier with the prefix F. And so this is one way of how those projects are addressing on naming their genomes. So we would like to identify similar best practices and also provide this as a recommendation for the AgBio communities. So, and then we have also for the locus IDs or the gene model IDs, we have different components again. So the idea here is how much of the information you can capture within the naming the locus ID. So here's an example with the Brassica napus project, where you have, since it's a polyploid, you have also a subgenome. It's so here is the C subgenome in green, and then you have the chromosome, the entity type here, the P represents the pangene, and then you have the numeric identifiers and also the annotation version. So one of the thing is, you can see also sometimes this naming convention can also get confusing when you come down to naming the transcript or the isoform, or if you want to include the version of the annotation and assembly without causing any confusion. So these are some of the points that we were addressing or discussing within the group. And so here's how, if you want to put it all together, you can see we get very long identifiers. And a good thing is some of them are human readable and they're accurate. And then sometimes some of the consortiums, they also use like machine readable ones which have a mapping file that you can always look to identify the different components. So Here we listed a few examples, and you can see from the different species and the different components that I talked to you about. And clearly you can see there's a lot of heterogeneity in this. So you can see the element order varies. So sometimes it's hard to distinguish which component is what, and also the conventions vary, especially for the species identifier. It can range from 1 to 3 like letters. And then there is also inconsistencies on what special or spaces or special characters you use. Some are use underscores, some use dots. But then, some of these, when you're actually processing it in a downstream database, it can cause a lot of parsing issues. So these are some things that, are not standardized, and this is something we want to provide as a recommendation to community and see if that's something it works out. So we thought we'll have a survey, and so our survey was targeted mainly to get a feedback from the community to address some of these challenges on the nomenclature. And part of the survey also was asking what are the existing metrics that are used for assessing genome assembly and annotation. So the survey went out within the AgBio community. We had around 11 respondents for the survey. So I won't give the results for the survey yet, because I think I had presented it earlier, but I'll give you in summary what the survey said. So it basically identified these most as the popular components for the genome and the locus ID, as we saw in the previous slide. And so from that survey, I think some of the things that we were thinking of providing as a recommendation and considerations for the nomenclature was, we could start thinking in terms of the larger projects like the Darwin Tree of Life project, which is, sampling around 70,000 different species, which also are sequencing and providing standard nomenclature. So this is gaining popularity. So they have this tree of life identifiers. They assign prefixes which are unique, and they have also a search where you can type in your species and get a prefix related to that. So that it is unique, but it has variable length, which is 7 to 9 characters. And then we also wanted to check whether having the variety or the individual name. So we need to decide how much what is the appropriate length to provide as a name for this. And then some genomes, they are polyploids, so whether to have the subgenome included or not. So that's another recommendations. And then the versions. So sometimes we usually characterize a version by like a single character, like a .1, .2. But over time, the annotations change. The word, it's incremental annotation, so it can go more than 9 or something. So how do we provide space for that? So here's what it looks like, the previous original IDs, and how when we transform it to the template that we have as a recommendation. So you can see, although we want to make it more standardized, and then have all those different components that are already what we came out from the survey and from the different projects, how we can organize this so that although it can they have different components, but then having it in the same sequential order will help identify those components easily. So here I think most of the examples here you can see are here are the commercial crops and other things, but then the identifiers get really long. So that is one of the things, but it's until you understand like what the component means, I think that serves the purpose here. So But then going forward, we also want to think in terms of pan-gene nomenclature, because I think as more and more genomes are coming out and the related annotations, there is a need to define what is a pan-gene. So I think the definition of pan-gene is also kind of evolving, and then now we have you can define pan-gene like having the gene that model that is present in one individual or all of the individuals within a species or within the same locus, and this can be defined between like having sequence similarity or based on synteny. But the also like how do we identify a pan gene? So there are the methods are evolving. So at this point, I think having permanent identifiers is a little premature right now. So, but that is something once these annotations and the pan-gene methods get solidified, I think we can focus more on some of those aspects. So some of the recommendations that we had within the group, also discussing with the maize community, was having this kind of clade specific pan-gene identifiers. So where we can have the clade, the version, and the pan-gene, the component for that. So yeah, so the last part of the of our working group was how does the community is using quality metrics to evaluate some of the genome assemblies and annotations. So as you can there is a lot of the genomes that are coming that are almost telomere to telomere, but there are some genomes that are still platinum standard, but like having gaps and other things. So there is a need also how we have a common catalog of these QC metrics that the community can fairly be comfortable using, and also evaluate the quality of the genome assembly and also the annotation. So, and some of the matrices that we had were at the time of where the technology was there, but now with the newer technology, I think there is also a need to evaluate the completeness of the genome. So, right now we haven't made progress much on that, but we had reviewed the literature and also reviewed what the larger projects like the Darwin Tree of Life or the Earth Biogenome projects are doing. So they have a good set of recommendations. So that's something the community can also adopt or use that. But now I think the focus will be if there is any newer metric that is emerging that can help easily evaluate. So that is something we actually want to discuss in the working group. So with that, I think our goal in the next 6 months is we still want to engage with the community and get feedback on some of these discussion points. And we hope to have a white paper done in the next 6 months and get that solidified. Yeah, we had a working group. We were a little shorthand at the beginning of the year, but we have newer members that have started to come in. And so I'm chairing this working group along with Sarah Dyer, who's the co-chair on this working group. And yeah, you can find more information on this or join a Slack channel. So yeah, we'll be happy to include new members and get this working group moving forward. So yeah, for the breakout session, so I think these are the 3 points that we would like to discuss. The nomenclature template that I discussed, the standard that I discussed, is something that the community would adopt, or is there something that we are missing or would like something the community would recommend? The other challenge that we had was how do we raise awareness of this nomenclature, and because there's every community, every sequencing consortium or every project has their own or nomenclature system, but there is hard to people to adopt to a newer standard. So how do we make aware or encourage people to adopt it? And also what's the target audience that we want to focus on, getting the feedback and also using the standards. And the last point we want to discuss, if if we develop a service as a part of this working group or some other project where we have a service that could mint your IDs, that is like, how do you encode or decode your IDs? Would that be useful, or would that be something that the community would appreciate, or it would help? So these are some of the points. So yeah, if you're interested in these points, please come join the breakout session, and I'll be happy to answer your questions. Thanks.
Thank you, Kapeel. We don't have time for a question here, so we'll catch up in the breakout rooms. I'd like to welcome Moira. So Moira Sheehan from Cornell University is going to talk about updates from Public Genetic Resources Working Group today. Do you like this one or the other? No, that one.
Hey, thank you. This is my first time doing anything with AgBioData in any formal way. So I brought forward some questions to get this working group started. And I'd like to kind of give you guys an update. And I apologize if it looks very different from what everyone else has done, because I'm totally new at this. Oh. Oh, it's fine. Okay, so we have a very small working group. Everybody on this call has been at least at one meeting. It has been hard to find time to get everybody together, and but Sunita's here and Jogi's here and Chougule also here, and they've been pretty much the constant members of this committee. So the first thing we needed to do was kind of define the public genetic resources that we wanted to tackle. So I'm presenting kind of the way I thought about doing this and kind of presented it to the group. In terms of the data types, we're thinking about data sets, marker sets, haplotype or allele databases, pan or reference genomes, and then biobank germplasm. And underlying all of these data types are data processing pipelines. And across that whole spectrum, some of these data sets are being held completely privately. They're not abiding by any fair principles. And then on the other side, in biobanks, you have a lot more of that clarity and fairness, but not much of it is private. So by looking at these things and knowing what other working groups we're working on, we had to admit that reference genomes or pan genomes and data processing pipelines are going to fall outside of what we think we can accomplish in a year. So we're going to focus on the last four that are shown here. The other thing that I haven't heard much of today, but I also missed all of yesterday, so it's possible I just was not here for it. I'm really trying to encourage our group to think about both fair and care data principles, since we're talking about public genetic resources. This has been an area where I've been trying to find ways to be an ally to Indigenous communities that have had their materials stolen or not misused and have not benefited from that. So whatever recommendations that I hope we come up with in this working group will really give enough space for care principles as well. Okay, so data sets really are multiple versions of data that are nested together in a relationship. In each of these large blocks, sorry, all my animations are gone, so I just have to point at it here. Can you see my mouse?
Yeah.
Okay, so each of these big lines here are multiple points when that data is being changed, manipulated, or altered as the processing goes on. Some data sets are closed, so additional data is being added at regular or irregular intervals. And although it's not shown here, metadata is associated with each one of these subsets and the pipelines that underlie those. So some of our questions are, how do we ensure to apply fair and care data principles to these different facets? Oh, that's not what I wanted to see. Okay, I made it in PowerPoint and now it's on Google Slides, so there's some compatibility issues. Next thing about data sets is combining data sets. Very often we are moving multiple different types of data together to make analysis. And they kind of fall into two buckets in our minds. There's the regular equally sized databases or data sets that are like things like weather data or sensor data. So years, time, days, hours, etc. Some fixed limitation. And then there's the irregular, unequal sized data sets, which pretty much covers a lot of what I work with my breeders. So trials, locations, sub-observation, genotypes, lab analysis, transcriptomics, metabolomics, and probably many, many others. So one of our questions that I think we want to be aware of is, do irregular data sets need different or more documentation than regularized data sets? Also, I want to talk about the next thing is marker sets. So every set of markers is a complete unit where the markers within that unit set define the properties and the usage. So these are set properties. And a marker set can be defined, predefined by arrays or pools, or they can be determined post-analysis as if from GBS. So again, we're trying to keep in mind, how do we apply fair and fair data standards to make sure that someone who picks up set 3 knows exactly what they're picking up and what kind of data should be coming from that. What net metadata must be associated with these to maintain future people using these data sets and knowing how to interpret that data. And so in this example, so all of these data sets all have 100 markers. Data set 1 shares 10 markers with set 2, 60 markers with set 3, but set 2 and 3 have no overlapping markers. But then what happens over time is these marker sets change. They shift and, sorry, now it's looking super weird. So anyways, pretend animation worked really, really well. So what happens over time is that these marker versions start to shift. So you remove bad markers, you add new markers, your assays get refreshed, but maybe the Vic and FAM fluorescence are switched to the opposite allele. And now you have different levels of overlapping markers. Still might be the same number, it could be totally different. So one thing that is important for a user who's using a marker set and then comes back 6 months later and doesn't know that the marker set is different, how are they going to interpret that? How do they know what's missing and what's not there and what's been added? And is there a way we could version these changes that provides that continuity of information in the metadata? That was a really cool animation. I'm really sorry you guys missed out on that, but super cool. Anyways, okay. The third type of data, public data or genetic resources I wanted to talk about was haplotype or allele databases. Because sequencing is becoming very affordable, even for crops or species that had never really had these markers, what we're starting to see is the detection of multiple alleles or haplotype in each marker. This is violating the SNP biallelic marker standards. So this is more like SSR markers. And with that comes the need to be able to keep track of all of the alleles that you're detecting, especially if you're using this information for breeding, because you're trying to find the allele that's giving you the trait of interest and move that specifically forward. So in our group, we're using DART-tag, which is a targeted amplicon sequencing, which does give you SNP calls, but instead it also gives you a small local haplotype. So we're trying to build these local haplotypes for each of the species we're working with, and then make that a public resource that other people can use so that the allele IDs are the same from user to user across that panel. So then you would have essentially for any species, a true allelic or haplotype database. And the last type of public genetic resources are biobank germplasm. So there's a push to use DOIs to function as a persistent identifier with interoperability with different banks like Genesis, PTR, in accordance with the FAO specifications. And so this is an effort that GRIN has been working on. There's already a GRIN global DOI document. They're not using the DOI field at this moment, but there are several organizations that are running GRIN global that are using DOI data in their database. So it's coming. It's just, what does it mean? So to really improve that interoperability with other germplasm banks, the addition of these permanent DOIs for GRIN maintain accession and or inventory IDs. So the inventory can be a packet of seeds, a form of plants and pollen. And in many cases, GRIN users are not aware of the inventory they're receiving. They don't know if it's a seed, they don't know if it's a population, if it's a true breeding line. This would give them a way to basically say, this was a population and I'm giving you an inventory of a certain set of double haploids from that population. So, trying to stick to my 10 minutes, I'm probably going way over. Our working group timeline is kind of ambitious. We're right here in Q2, but I think we've settled on what data and resources really qualify as public genetic resources for this working group. And we're currently working on what are the limitations to care and fair and for access to public genetic resources. And one of the things we did think about was whether animals or plants resources had to be treated differently, but in the end, I think it's more about genome size and complexity. So it's more about like, how do you deal with polyploids, like autopolyploids in JBRUs? That's not really been addressed. And then we'll work on trying to figure out what would be acceptable solutions to fill the need and determine if any of the current publishing options could be modified, amended, or need to be created anew. And I'd like us to propose a solution with the decision tree of how somebody would navigate to know how to provide the proper metadata for their example data, and then present these findings back to AgBioData. And that's it.
We have time for 2 questions.
This is totally maybe of this here, but I'm curious, who is doing the DOIs for GRIN and or is someone issuing the DOIs for all the germplasm repositories as part of that?
I have no idea who's doing that, but I know the National Ag Library is also considering doing DOIs for data sets, but they are still in working group.
And then the other thing is I would say that if I was looking at like where there's a huge amount of overlap, the genetic variation working group is really looking at assigning stable identifiers with DNA, and RSIDs are very common things that are used within human in the medical area to, so if you go to 23andMe, you're getting an RSID, right? And so this, I think, would be a natural area for thinking about where those working groups could think about looking at data.
Yeah, so after we talked last week, I went and looked it up, so it's on my radar now too. And I'm going to be working with anybody's welcome. We really would love to have other people who have thoughts on this type of data. If you're interested in joining, we would love to have more members. Hi, this is Megan. I'm curious what your outcome for the fair and care step is gonna look like. Do you think it'll be recommendations, a summary? I think what I'd like to do is make sure we're not putting up the new barriers. I don't want to be the person who's telling somebody else how to implement fair. I just want to make sure there aren't any roadblocks to someone doing. So if someone's going to use blockchain to do federated data calls, then I just want to make sure that we're aware that that exists and make space for that and find other ways to make data accessible. Even if it's being scrubbed of some of the metadata for privacy. Okay, that's what I was wondering. Sorry.
Do you work also with like CIMMYT and like ASAP or other, are you thinking of when these are just US based?
I don't think it matters. I think it could be anywhere. I would hesitate on making recommendations to other seed repositories because their needs may not be the same. But I think in terms of the publishing materials that come out of those, I think having a DOI of knowing what accession you actually use would be very valuable for someone to replicate somebody else's work or to take that work and move forward.
Yeah, I've seen DOIs given by agencies, and also IPK.
IPK.
And yes, yesterday during the breakout, like we were talking, we need to interact a lot more with the standard for genetic variation group and your group. Maybe we'll invite you over
Yes.
to some of our meetings and.
Okay, perfect.
I think one last from Nina.
This is just a really quick comment or a question about, like, in the space of, permanent identifiers, like, is a DOI the thing that you want, or like, what about, like, was it the RRIDs, the registered resource ID? So there's a whole other sort of like namespace for. That would apply to things like strains and germplasms and stuff like that, that's in use in other areas.
Yeah, I don't really, I mean, I guess I don't really care what the vehicle is, but what I'd like to see is something that is nice and short, like a URL that can be dropped into publications on website. And when it changes, there's a versioning, some way of tracking the version. So this marker panel of 3,000 markers has had an update. Now it's version 2. Here's the new DOI for that. So that whenever you're, this is also for vendors who are sending you the data back to make sure that they send you the proper metadata back so that you can use that data. So that's a problem that we see on the horizon, but it isn't here yet, but it will be. If at least in my group, it will be here.
Thank you.
And Rita?
Moira?
It's just a suggestion, like, because you missed it yesterday's session. I think this is, so I think you missed like the presentation of the phenotype working group from yesterday. We will post the recording, but I think this working group kind of trying to solve one of the limitations that we posted, that is like missing an ID that can kind of combine all the information that you have for a specific data set. Okay, and so just like
Thank you, everyone.
So we will start with the report from, so who is reporting? The in-person breakout room and the genome nomenclature working group that is only working. So the first one reporting is Jennifer Platt for about the breakout room for the Federation thing. All right, thank you.
Perfect, okay, so, our first question was, as a database maintainer, what types of data you have available that you think is in common with other databases or systems, and what do you have that's unique, but very useful? And the reason we asked this question was because, for data federation, we recognize that a lot of our databases likely have data types that are very similar and could be easily federated. But there are maybe some databases that have unique information that everybody wants to access. And so we want to make that available through federation. But it's not something that we want to replicate, or ones where we're going to pull the same information from multiple sources. So we talked with the genotype and phenotype working group, who apparently has this phenomenal, very large Excel spreadsheet. It's Excel, that has all these databases for plant and talks about what data they have and how it's structured and what are the required data and what's, so we're like, we need to get that, and we need to get their white paper. And then, I wrote, we should reach out to the FANG group to get similar information on the side, because that database is for only plants. We did not even get to community standards, so that's why I crossed that out, not because we don't like standards, but because we never got to that part, so I was like, we're not gonna talk about that. Okay, next question was, as a user, what data are you having difficulty accessing? And what we learned was the biggest challenge is terminology, that different databases may call something a gene or a gene model, and it can be difficult to figure out what maps to what. Somebody suggested we need a thesaurus. And we also had somebody talk about doing variant calling, and sort of the way it works is sometimes you have to go to a database and then you have to upload your file and then you get something done, and then you have to download the results. And there's a lot of moving large amounts of data back and forth and back and forth. And what we would really like to get to is where you don't move your data and you get the tool, right? So the tool enables you to pull in that information, you have it locally. You don't have to send your data to different databases. So that's what we recognize as one of the challenges. It's not necessarily maybe type of data you're having trouble accessing, but the way you're accessing it is very resource intensive. So maybe we need to work on that one. Okay, next question. What medium would be best for the training? Today's training day. Thank you, Jason. So what types of repositories? So the education working group talked about some of the efforts that they're doing. There was a request for sort of just-in-time training. So if you're at a database and suddenly you forgot what you're trying to do, or you don't understand a certain term, could we embed little trainings into databases to sort of help the user to navigate? We do pre-recorded videos, and we talked about how we would present that material, that the audiences may be different. I'll get to audience next, so that's why I haven't mentioned audience yet. But we did talk about doing videos, and we wanted to do a demo. I know our working group is training, but part of training is seeing and actually walking through a use case where we go, oh, we've been talking about data federation. This is what data federation looks like when you try to do it. And so we got, somebody suggested that some of the hardwood databases have done a federation use case, so we could use that. We had a discussion about primary repositories where really you keep the raw data versus secondary databases that already do a certain level of federation, because they'll take the raw data and then they'll go, ooh, you would also like to add with this information. You would also like to link this to this information. And so different databases sort of have different purposes. So we should talk to some of those secondary databases that already are pulling in information from primary sources. Last question. What is the audience for the training and who needs to be trained? And what we recognized was that the education working group, their audience is primarily students. And that for the data federation working group, our audience is more the database maintainers, PIs, developers. So for example, you're a developer and you want to create an API to do task X. What is the best technology, right? What is the best way to build an API that will be maximally useful and efficient, right? And so it's a different audience than what our education working group is focused on. And so what we would like is training on skills and technologies that will be enable you to do something. We had the comment that a lot of databases have database specific trainings already, right? So you go to a database and they're like, go to our learn page, right? And there's a ton of stuff, but we might lack training that's database agnostic, or I want to do this, and how do I do it for databases in general? What is best practice? Right? So we thought we'd focus on that. All right. How am I timed? I only have 5 minutes. So, good?
Thank you, Jennifer. So now is the time, so it's reporting from the education working group. I believe it's Megan.
Hi, everyone. So we had a very robust discussion and didn't always stay on topic, but that was good. So just to kind of summarize the working group very poorly, is that we're looking to try and educate students on the fundamental theories and services needed to understand how the GDB databases and services work, so that you, the student, can then use them better to do your work. And so this is not how to use a particular service, it's how to, like, what are the fundamentals you need, theories you need to learn to do this work. Because we have a lot of, it was just explained how to use a database, that's fantastic, but you know what a database is. And so we talked about like, what do we need to do to get this in the hands of the educators and other people to make it useful? And so one of the things we got back was YouTube. Everything needs to be on video and online and easily accessible. So that's something we're definitely going to look into doing and implementing. We do need a review and feedback mechanism of the materials before they're finalized. Jason, this is where I follow up with Jason's smiley faces. So thank you very much for helping. And the faculty mentoring network was mentioned because they provide mentoring for lesson plans and implementation, which I thought was really neat. I hadn't heard of that before. We also had PS integrated ontologies because that's one of the lessons that got dropped because we kind of need someone to kind of champion that and write that lesson plan. So if you know anyone who can help either write it or point us to materials and stuff, please do send us feedback and information. You don't have to maybe contribute a lot of time. We just need some help with it. So question 2 is kind of how do we get the word out and what comes next? So YouTube, I could have been on this slide too. But the main question, is that we're making this thing, we want people to use it, how do we do that? And so we have a lot of professional societies we can reach out to. We have some listed on here by name and some I just kind of had generalized. We also think that this material right now is focused mostly on plant databases because that's the strength of AgBioData, but it could be adapted to other groups as well, like the insect bioinformatics. And we need a better communication plan. We need to be able to make it easy for people to join, and also invite people in to be able to get our information out in different venues. And then we should also consider formal publication and peer review of those materials, as well as just like informal review and stuff as well. So I went through that very quickly. And I think that's it.
Yeah. Thank you so much. And so now I think it's time of the Genome Nomenclature working group. So I will stop sharing. And I believe there's Sarah from Sarah Dyer online. Sarah, are you there?
Yes, I'm here. All right, can you see my slides?
Yeah. Yes.
Great, thank you. Okay, genome assembly and annotation nomenclature. We had a really nice breakout room with a diverse group, so that was really, really good. We kind of took our notes on here, so it's quite, it's a little bit detailed, but I'll whiz through. They're not all so long as the first slide. So we started off by asking, would anyone who is joining our group adopt our proposed nomenclature system? And we got some feedback about the annoyance of locus IDs not being a fixed length, which is partially due to the fact that the species identifier that we would use if we use the Darwin Tree of Life ID system is that's not fixed length to start with, and then also varietal names tend to not be, and we're aware of that, but we're also not sure there is a solution, so that's why we've kind of gone with where we are. But we had a nice discussion around there, and some other features like the numeric IDs, that how people like to kind of number them. What was really interesting as well was a discussion because we had Tanya from the Arabidopsis kind of from TARE and talking about, there we sort of had a contrast of people with thinking about new genomes being made and thinking about very existing references and how this might map to a new nomenclature system. So that was really helpful. And we hadn't considered a community consensus type of an assembly. So we need to work out how that would fit in the ID. So that was great. And then we talked about how these IDs might be used in publications, because obviously they're quite long and unwieldy, but they're also very specific, which is the point. So therefore they ought to be used in publications, even if you then can kind of abbreviate them in the text. So we sort of had a bit of a think around there. And we got into Busco a little bit, how they have a kind of an identifier that then works and kind of feeds into the quality and annotation aspects. So in summary, we had some positive feedback on the adoption, but sort of more work needed, which is pretty much where we knew that we were. So we will kind of be reaching out as this continues to develop, and we'll be looking for more feedback from you all. So that was our first question. We talked about how to raise awareness and encourage adoption. And so, we've done surveys. We're thinking of producing kind of like a paper on the standards, and we need to put that somewhere where it's broadly seen by our target audience, whether that's purely agricultural, kind of AgBio type genomes or more broadly. We talked about whether the big repositories would need to be aware of this, and whether they would enforce it and how that might work. And that's something to be explored, because if this is a standard that's only for AgBio species, then that's sort of a bit tricky for general purpose repositories to sort of work with, we think. So, but anyway, to be explored. And we talked about the fact that when you're submitting something to your like community repo, there will be curators there that understand what the standards are. Whereas if you're submitting to a general journal, the editors wouldn't necessarily be aware. So it's important to make journals aware of this if we want them to kind of enforce the adoption or encourage, however strong you want to say it. But having Daniela there from Micropublications was great, because we discussed a bit the kind of loop you go around with the working group there on the FAIR data, and how we can join up with them and test things out a little bit in practice before we sort of take it more broadly. So that was really helpful. And the third question was, if there was a service for this, would you use it? So of course, the big question there is who would host and maintain such a thing? And there were questions around who is the authority to issue names. For model organisms, there tends to be an authority. For other species, there are not. Everybody names their own. So how might that work? And how much metadata would be associated with an ID, so people could kind of get back to all the information, the more full information. So a service could kind of provide that, but it's important. And I've just grabbed your thesaurus word that came up in the previous talk, because that's sort of what we're talking about here. And if you had this kind of a service, could it be integrated into like repositories? So like knowledge bases. So if you're in a repository, can you then query this service and use it for your search without having to kind of leave and do the searching somewhere else and then bring that information back to search in the resource that you're in, right? So we can kind of integrate it. So we thought that would be very cool and useful, but we need to find someone that would host and maintain it. And we asked for contributions, and Daniela and Gerard are kind enough to volunteer to help us. So very good.
Thank you, Sarah, and all the people that are holding online. And so the last reporting will be from the Public Genetic Resources Bureau, I believe it's Jackie.
Are you watching? It's okay. We're still close.
We thought you were done with me. I'm back.
All right, yes, I do have this normal energy. I have two kids. It's not good. There we go. So I have to summarize some general things. The first question was, what are the blind spots that we're not considering for public genetic resources that we have chosen to address? So let's talk about the elephant in the room. There is a lot of blind spots and high privateness of data, and there's not much we can do about that. We can encourage, we can say, "Hey, let's share, let's play." Thankful. Let's think of something that we can focus on. So like, how about the genome, the germplasm from public breeding programs? How to really document the exceptions that they remove from a program that they don't really want to continue using. What about the pedigree? Can we get that information at the very least? And then a university breeder talked about the option of actually even just using their university library archive to get the DOI, to put that information available. I mean, because where else was the person going to put it? And then we also have to think about haplotype ALL databases. Like, we need to be able to share data and think about like, how do we consistently share data with the right, consistent names or in the same location within the same population and various things like that. Future actions. There are no standards right now. Courage, like, let's share, let's be nice. Encourage, good practices. start where, what is released to the public, you know, really start getting good IDs or DOIs for what's released. And public databases, there was one idea that was brought up about like a public database with a sequencing vendor. So basically it was available for all breeding programs to see the presence and absence of what they had in their breeding population. And they can continue, they can, you know, even the private and the public can use this data to see, hey, what's there? And if they wanted to add to it, they could, and it would kind of help to see how is there in general, their breeding population to others. And of course, encourage more engagement with the National Ag Library, because this is something that we really useful for it. Next question, what is the minimum amount of metadata required to accompany each type of public genetic resource? And is there any way metadata can be verified before publication? There is a template available, but the person can enter whatever they want. As long as there's something in the field, it doesn't matter. Someone needs to verify it. We also have the general problem of old breeders and young breeders. Old breeders, they don't care. I'm set in my ways. I like my lab book. You are not. You're taking this lab book that I write it from my cold dead hands. It's exactly what one soybean breeder told me. I'm like, okay. But the younger ones might be more willing to kind of work with us. So kind of trying to teach them, you know, why is this important? And really work on, you know, this is difficult for them to learn, so when we work on some training. So that was one of the things for our future actions was, you know, getting the data curator in there from the beginning to really work on that. And then the basic training. We've been talking about this over and over again. We're seeing a kind of continuation of this. How should genetic resources for autopolyploids be handled to best suit users? This was a topic, and you thought like maize and soybean was hard. Like for there's an example for this, because I'm like, what does this? And what doesn't act like a diploid? Alfalfa, blueberry. Apparently, there's a mix in roses. That was amazing. You know, basically everything that we have developed is for species that act like a diploid. Nothing has really been built for something that is an polyploid. So basically, we need to build better data representation tools for polyploids. And that basically means we need to talk to these breeders and these researchers to see, to understand what are they working with, what are their problems, what are their challenges, and how can we even assist them in the first place. And the last question was, how can we implement care data principles into our recommendations? Basically, it was like just a simple, like, okay, the problem is I have no idea how to approach these people. I need to be sensitive to, you know, everything that's involved with this, because there's a lot of questions. Who owns this entity? We don't want to spread, you know, anybody's feelings. Need to be very careful. There was mention about a biocuratable label that was associated with the genome, and then like it required that a statement was put in any publication about it, about, you know, where exactly this came from, and who owns it, and various things. So it's kind of like there. And the idea probably to focus on this more is really to implement a care principles working group to really think about like, how do we interact more with native species type of things, because everybody thinks about, you know, those wild genotypes we needed to bring back in the population, but we need to be careful where we're going. So, any questions? Oh, no, I have to go on there.
Thank you, all of you that have done a great job with our role. So the last but not the least presentation. So we will have some closing remarks from Soup before lunch. So it's very short, don't worry. Okay.
With one slide actually, and so I just like to thank all of you. I enjoyed a lot, and I got to know people and learned a lot about other working groups. You guys are doing fantastic jobs. And we also, our working group got great input, so it was, I think, very useful, and I hope you think the same. And so in-person participants, and also like I'm very impressed with the online participants working really hard. And I just wanted to also like thank Anarita. You've been like. And it's their full funding. And then. You got it. How about the motor? Okay, do you want to do this or? Okay, yeah.
Okay, so please for the presenter, if you can, so give me the permission to share the slides online, so we make PDF and put online, so please, if you are against, reach out to me and say that, but I think it will be okay. And I will send the evaluation survey for in-person online people in the next day, so please take it. I know we are very short, but please take it because it will help us for organizing the next events of AgBioData. And so you are welcome to edit, use, have more feedback in the Google Docs that you use for the breakout room notes. So I'm sure that you get a lot of useful feedback to implement in the following month. And so I will follow up with you, sending more instruction on how we will ask for reporting in the year. And yeah, so okay. Oh yes, so there are also the online notes, the breakout room notes from online people. So I will also share this with you. If you don't have access to them, please let me know. And with this, we want to thank you for coming. We wish you safe travel, but before travel, there is lunch in the other room. And I want to remember, if you haven't done it yet, book your shuttle to the front desk. And that's it.
Thank you.